presenting to y'all. I like to sort of describe some of what I do as multifractal social psychology, not like traditional social psychology, but I see groups where others don't, and so I've sort of taken to seeing multiple people as well as single people as well as single organisms as themselves swarms. I got into this for reasons of things like embodied cognition, and that sort of led me to start looking into this cascade metaphor for what fluid systems are doing when they're engaged and embodied in their surroundings. So So a short version of embodied cognition is sort of like is the idea that we definitely have to have something on our minds before we act, but the action is very important. So we do have these old plans, this old information, but every time we're leaping out into the world, we sort of stir up new stimuli, new sensations. We also are always getting new information every time we move. So we sort of we act to think, and we don't just think to act. And so this idea, I think there's a lot of ways to take embodied cognition, and there's a lot of ways to sort of think of, for instance, the body as a puppet of the brain, and sort of the brain sort of greedily collects all the information, but uses this interesting shaped puppet. But I think that it could be a little more fluid and a little less sort of centralized, and it could have a lot to do with sort of how the fluid shape of our actions are impacting how we learn about our world, and so the body might be playing much more of a central role than sort of traditionally thought in psych. So all these sort of interactions that a body has with its surroundings, they unfold at multiple scales of time, and so according to the math that I've seen, at least, it's sort of that's led me to sort of suggest that these shapes that our movements take are cascading, and you can see cascading forms throughout our physiology, but I want to sort of see how far that goes through our actual measurable behaviors. And the cascading shapes, so far as I've been able to see, invite a multifractal geometrical model. So if I want to understand what we're thinking new based on how we're acting, my approach has been to sort of look at the shapes of the movement in terms of multifractal geometry. And I'll be unpacking some of what that means. But the idea here is that the multifractal shapes of this sort of swarm of bodily movement can reveal interactions across scale that underpin what the swarm can know. I've started with sort of single organisms, but I've sort of gone into other model systems, multiple people, but also other organisms where it's much easier to see a swarm all at once. And the swarm metaphor is a little easier to sell. So it's hard in psych to sort of portray an individual organism as a swarm, because so many people are sort of invested in this idea of all events sort of going towards the service of the central nervous system, of the brain. So what I'm proposing here is a multifractal route to swarm intelligence, where multifractality in what follows is operationalizing the interactions across scales that I think, and this is where I'm sort of still learning, still sort of taking a lot of feedback of my own through my own steps. I think this characterizes swarm intelligence. And I think this is one way to understand how swarms can act adaptively. So that's sort of the opener. I do welcome any questions along the way. So I will try to pause at the end of each slide. If anyone has any questions, I'm happy to take them. So what's the best way for me to, I guess you could just call out, I guess, if there's something, and I'll happily zip back. Okay. So any questions? No. Okay. Well, so one way to sort of start digging into this idea of this cascade is to sort of sort of schematize a body in action. So we could think of lots of these. I mean, so the brain does matter, and it does matter for setting off these sort of these massive chains of progressively smaller events. So you do get this sort of groundswell of activity in the brain, and that can sort of have propagating influence down a spinal cord through flexors and extensors of muscle pairings, and the muscle activity in turn induces the fluctuations onwards to the periphery that are actually, so given the speed of electrochemical transmission, these sort of distant body parts are actually so far that the brain probably doesn't sort of specify most of what they do. So much of this periphery is sort of acting relatively independently, and it's being coached along by these more central aspects. And so these fluctuations, crucially for cascades, are spreading out anywhere into progressively smaller fluctuations. So they just sort of, they're dying out, breaking down, but they keep spreading. So there's not necessarily a principled center. So we can start this conversation from the brain, but we can also sort of moor this organism in a context that's full of important informational gradients, such as time and clocks and tools and the ground surface. And so each of these parts that I've circled can be sort of key players that end up reshaping this fluid cascade that sort of is rolling around amongst them. And so what we get is this intricate spreading out of fluctuations that are essentially turbulent. So they're these turbulent structures where these large fluctuations can sort of beget smaller fluctuations, which then in turn can sort of scaffold and constrain the ongoing large fluctuations. And so we do have, I believe it feels strange from psych to sort of start talking about how we have turbulence here, because I definitely don't have a physics PhD, but it looks like what turbulence is in all the models that I've been able to understand. So that's sort of, so I come to this through sort of the psych subfield of movement science, where they have a big problem of understanding how a single organism can move all of its degrees of freedom around. And this is sort of an ongoing challenge as to how it could be that all these different parts get moved around. And so one way it could be is that we have these emergent synergies that are organized hierarchically. And so I try to portray that like so. But any questions?
So where do central pattern generators fit into that scheme?
Central pattern generators, they remain controversial, for one thing. There is no necessary need for anything to be devoid of its own resonant frequency or its own timing structure. But in order for the whole organism to work, you do need a fluid hierarchical organization that central pattern generators can participate in, but they are not sufficiently global to show the organismic coordinations that we get across the whole body. So we can sort of envision this cascade across different time scales as well, and that becomes crucial for the analysis that I'll be using. So we could think about this intention to run as waxing and waning across time, and that could be a relatively long time scale as we're sort of rushing through an airport, say. And so at the longer scales of time, there is the more slowly changing surroundings, which is sort of our airport gate full of concessions. And so you might slow down if you're suddenly feeling hungry and you smell something tasty and you're looking around for where to go. And so all of a sudden you start triggering these sort of medium time scales that sit within your long-term goals of getting to the gate, and you're sort of ready to sort of compromise, and you start exhibiting all these sort of finer scale fluctuations where you bring online new physiological events as you sort of take a pause and your physiology kicks into gear, sort of investing in this distraction from your long-term goal. So you have these very fine short time scales, and then you can sort of get sort of kicked back into gear by another cue that sort of reminds you of where you have to go, and then you might not have time, so then your adrenaline starts going. So you have this ongoing cascade, and it can eventually bring you to a happy conclusion. But so as a movement scientist, sort of unpacking this choreography, the trick is that there's no sort of quiescent time scale. There's no sort of, so you don't get anything that's sort of quiescent. It's a pretty thoroughly unstable, non-stationary system, non-ergodic as well. So as we consider ever finer scales of time, we don't actually stop finding variability. We just uncover more subtle variation. And so you can sort of make your whole successful research program based on any one of these levels. But the thing that keeps me up at nights is sort of how to express how these things interact across different timescales. So how do we contextualize any of these so that they help us get a job done? And so for me, that's where multifractal geometry becomes most useful. So any questions about this layout of timescales and the interactions among them?
So what are you measuring? The jitterness or the trough and marks?
So I will be getting to that. This is just sort of a schematic of sort of how we are a multi-scaled system that has multiple observables within it. So I've measured many of these things. The intention to run is sort of a schematic hand-wavy thing to sort of relate over the idea that we do have a waxing and waning and how we're engaged with the task. Many of these other observables, you can take any single one of them, and the catch is that each one of them actually shows an imprint of all these different interactions. So you can care very much about which you're interested in because they each reflect a different point of contact. But actually, you can also find that all of these things all these sort of one-dimensional observables will actually carry the imprint of these complex nonlinear interactions across scale. Okay, so schematically, again, this is just sort of what we're doing in fractal analysis is we're taking a standard deviation across multiple different timescales. So really, fractal analysis sounds sort of newfangled and weird, but it is actually sort of just a generalization of standard diffusion modeling, where you're looking at the relationship of standard deviation with timescale, and you're estimating this alpha here, which is an exponent there. So you're using usually logarithmically scaled axes to estimate that exponent as a slope. And so the interesting thing about these relationships is that the logarithmic scaling sort of makes plainer to the eye the fact that there is this scale invariant relationship that you can find across multiple timescales, and the power law is sort of a mathematical indication that these relationships diverge and don't converge to any sort of asymptote. So what we are doing here is estimating that slope alpha to estimate the exponent on the left side, and that exponent is effectively a dimension, and so. And if you had a Euclidean system, you could look at these exponents and get from them the standard Euclidean dimensions that we know and love. So what this will do for us in what follows is sort of answer a question about how strong relationships are across timescale. And so we can use this operationally to sort of test the strength of nesting relationships as they support perception and action. So, and anytime I say multifractal, which is often, that is just to say that this organism is not like a monolith. It actually embodies multiple of these fractal scaling patterns. And so, in effect, any one of these observable time series actually has within it multiple scaling relationships, and this has been in the works operationally for about 50 years. People have been doing this kind of modeling, and it often shows up in like geophysics and meteorology. where they're interested in sort of turbulent flows in the weather or in fluid flow. Any questions?
I have a question here. Can you extrapolate this? And what would probably the next higher order of organization be? Or does this somehow resonate with something in society, like a group or that way?
Yeah, so I will be making the case in subsequent slides that this capacity to find scale invariant relationships and sort of describe them with these exponents here, that it does actually it does seem to resonate with or predict group memberships or capacities for different parts of the body to coordinate with each other or with the task environment. So yeah, so the scaling up does go somewhere and does become, I think, useful for talking about the social kinds of coordinations that go on in any kind of swarm-like system.
Cool, thanks.
Great. So I'm sort of also working from a biological standpoint, which is very systems-ish. So in developmental psychology, starting in like the 70s, they started to talk about this framework called probabilistic epigenesis. And this is sort of like the epigenesis that is more sort of plainly in the textbooks, but the probabilistic adjective here was sort of, I think, the developmental psychobiologist trying to understand how these could be sort of less deterministic, more random. And so they started trying to formalize these ideas in terms of these multiple layers, all of which are interacting at all these different scales of time. They're going up and down the scales. And so you have sort of multiple layers, and some people would call these cascade models and some people wouldn't, but I was always sort of seeing in this schematic some sort of glimmer of what the physicists were talking about in terms of turbulence. So all these participating variables, crucially, are not just sort of like solid lines, they are fluctuating. And so you do have sort of these relationships that can wax and wane. And so I've been sort of, was sort of one of my inspirations going forward to sort of try to see how we can elaborate this idea and sort of think about it in more fluid terms, where we could use multifractal analysis to model the exchange of cascade dynamics across a coordination, which could show us how symbol-like structures can emerge or dissolve, and sort of individual selves or others could also evolve fluidly within the same swarm. And so we could sort of elaborate that idea in sort of social biological terms as sort of an overarching social context, which would sort of serve as like the environment, or as a part of that environment. And then an identity sort of forks out of the relationship of a body with its context, or a component agent within a cloud of agents, for instance. And then you have the behavior also sort of spreading out of this body-gene interaction that's also scaffolded by the context. And so you sort of, I think in practice, the traditional schematics here about sort of developmental systems is actually a lot more messy in practice if we tried to like sketch that out. So that's what I'm attempting down there at the bottom of the slide. And so I sort of wanted sort of a way to think about sort of how we could sort of treat swarms and fluid coordinations within a single body as this interesting cascade model that psychologists had been sort of tinkering with for a while now. So how to unpack that? I wanted to sort of take that idea and apply it to this idea of a multi-degree of freedom organism or system as it can become a swarm of several organisms. And I wanted to take that sort of biological spider web there. And so Gilbert Gottlieb, sort of the founder of this particular schematics idea. So he wasn't alone in this intellectual stream, but this idea of probabilistic epigenesis. He was not a mathematician, so I never got the chance to like check with him, like, how do you think this resonates with what you're doing? But he I've heard from scholars who worked with him that he wouldn't have minded my particular blend, which for the time being has been vector autoregression, which is a sort of a very bare bones causal model that allows you to sort of fit short lag relationships between multiple contemporaneous variables. And then it sort of fits sort of the single variables relationships with its future selves. And then it's also able to extrapolate the effects of each of those participating variables on each other at similar lags. And using that has been very useful for me to sort of understand how we can take multiple measures of the same system. And so we could look at head sway or hand movements and eye movements and sort of put it all in one sort of family of endogenous variables, and then imbue that with multifractal fluctuations, where what we're doing there is we're sort of, the participating variables are the estimates of multifractal structure. So this could be in a way to elaborate these sort of systems bio perspectives, such as probabilistic epigenesis. And so in that way, we could sort of imbue this schematic with a little bit more fluid structure, kind of like what you would find in a turbulent model of development. Any questions about that? All right. So we can sort of do that. So the actual math of this is to look at standard deviation and look at how standard deviation grows as a function of time with that exponent alpha. Now we can apply this to heterogeneous systems where you have multiple different parts, multiple different sizes of events processing at different rates. And so you would get different kinds of diffusion within the same organism or the same swarm. And so you can sort of elaborate the fractal analysis on the left for heterogeneous systems on the right, where you can basically take that initial time series on the left, and you can use this Q filter to basically accentuate small events and crush out big events. And conversely, you can sort of increase that parameter, so the other side of 2 there, and you can emphasize large events at the expense of smaller events. And so you can basically take a single time series, and so long as you have heterogeneous events, heterogeneous sized events, you could potentially get multiple different temporal profiles. And so see that large events are evolving differently than smaller events. And you can estimate a fractal parameter for each potential event size that you could estimate. And so what you get, as I was pointing out before, is you get multiple of these scaling relationships. And so you get a multifractal generalized alpha. And now most fractal analysis will estimate that alpha for a Q of 2. And so much of what follows is sort of asking the question of, could different size fluctuations actually show an influence on one another? So could you actually find that large events and small events are buffeting the intermediate events that have been emphasized by monofractal analysis? So generalizing this exponent allows us to sort of talk about how large events or small events could be bumping up against each other, and whether or not those relationships matter for intelligence of the human organism swarm or of multiple organisms together swarming. Any questions about that analysis? And so the vector autoregression is serving the role of testing whether or not these different event sizes are actually influencing one another. Okay, so I'm a psychologist by training, and so one of the sort of things that we study there is executive function. And so this involves the ability to selectively attend to one of many dimensions. So we gave people a card sort task. So a card sort task involves sort of having the participant look at a number of cards out of a deck, and they have to sort them. And so the rule can change as to which attribute they should be sorting by. So animal, ornament, or color, for instance. So they might get the purple cow with the glasses, and the question is, which other of those three up there should they sort this card with? By color, by accessory, or by animal? So participants were randomly assigned to one of two conditions. One condition, they only got an explicit instruction of the rule, and so they would be told, you have to sort by animal, and then they would, sort by animal. And then in another case, they were asked to induce the rule, and this was slightly more challenging. So they had to sort of figure out by feedback which pile they should be sorting into, and then the rule would change. And so we would ask them to have 10 subsequent correct trials before they had mastered the rule. And so we looked at hand movements. So we asked sort of what sort of structure would there be in the hand movements used to sort the cards that might give us a window on sort of the flexibility of the cognitive system as it moves through this task and as it learns to master a new rule. So the hand movements, we took three-dimensional hand position measured at 60 hertz with a magnetic motion capture system, and we sort of took that continuous movement and we used a sliding window to basically estimate many different fractal exponents across time. And so what we found is that if you looked at these windows, these epochs, then you would see that in the explicit condition, on the way to mastery, participants would show hand movements that basically defractalize, to coin a word, on the way to mastering the rule. On the other hand, if they had to induce the rule, we saw this interesting parabolic shape. We had seen this in another task using an insight problem and found a similar path, basically, that having to sort of figure out the rule on their own seemed to occasion this sort of this inflation and then decrease of the alpha. And so for the purposes of the multifractal swarm question, we wanted for our next step to ask, well, are the changes here that we're seeing that we had seen in other insight problem type tasks, is this due to the contribution of multiple sized events? So is this person, this hand movement, is it buffeted by events of multiple different sizes? And is that important to the trajectory that we can see so reliably in the hand movements? Any questions about that? Okay, so you can basically think about this, the multifractal generalization of that earlier point in terms of this sort of manifold, where you can see Q on the left is this dimension that can basically change which size event the fractal exponent is emphasizing. And then on the right, or the horizontal, you have epoch, which is sort of the time in the task. And so the fact that we got this drop off on the vertical axis was evidence that the sorting hand motions are multifractal, and there was this decreasing alpha with greater Q. And we sort of can situate our previous slides finding on the manifold like so, with that pale curved arrow. And so you get this drop off at the end. So we use vector autoregression to basically test unique effects among these bidirectionally interacting variables along that multifractal manifold. And so we had wanted to understand these two relationships here on the bottom right. And so we wanted to know whether alphas for in different parts of this heterogeneous system were contributing to this. So if the body was a cascade, then we should find this interaction between large events and small events. And so we basically wanted to know, would fractal parameters from before lead to later changes at the Q equals 2 alpha. And so what we found using vector autoregression was that indeed there were unique effects of the alpha at q equals 4 and the alpha at q negative 4. So for large events and small events respectively, they both in the induction condition evoke later increases in the alpha at q equals 2. There was a condition effect where if we just gave people the rule, then the effects within that cascade seemed to be the reverse. So the alpha for large and small events evoke later decreases in the observed fractal shape. So what we were confirming here was that you don't just sort of have a monolithic fractal system that somehow resets across time and sensitively to task constraints. So the task constraints somehow speak to the moving hand through a cascade of events where these large events and small events, which you can estimate separate fractal dimensions. These different sized events showed a different pattern of interaction within the same movement system, and that was evidence to us that it was sort of beginning evidence that we had of cascade dynamics that would change based on task. Any questions about that? Okay, so then we're sort of going to move from this executive function task into some other kinds of tasks. So here we also looked at coordination behaviors. So if you're interested in movement systems and movement coordination, you might see a lot of research on timing behavior, so the ability to keep time. And so you hear a beeping metronome, and eventually you start encoding the regularity, and you build up an internal model of when to tap next and start anticipating the next beep. But so then we started to ask, what would happen if you made those intervals random, if you made them actually mathematically chaotic? In that case, it's mathematical. I mean, so if you're coming up with a prediction based on any sort of linear model, you're gonna fail at this. So it's linearly unpredictable. So we built this using the Ikeda map, and here's what an example metronome interval series looked like. So as you can see there, the interval durations were scaled to hover between about 1.5 and 1 second, and there was some variability within that range. Here's what human participants looked like. So these are the intervals between their taps, and you can see they're in a good ballpark. They're following along. So their task is to tap along with the onset, so not after or not before. Try to keep in time. And so this was very challenging, and it didn't sort of submit to a lot of the standard decompositions of this task in the rest of the literature, because sometimes people would omit to tap, they would tap twice, they would sort of, so it was very difficult to know what to do with this. What we found initially was that the fractal scaling actually matches. So the alpha of the human behavior seemed to match to sort of tune to the task. And so this was sort of, at least sort of in the absence of any predictability, it seemed like, okay, so these fractal bodies seem to be able to however they were sort of increasing or crushing their fractal scaling in the executive function task, they seem to be able to absorb sort of environmental structure. And so you could think of the human and the metronome as like a very tiny swarm, where the human is trying to absorb the information from the metronome. And it's a one-way street, but it's an interesting one-way street. So the initial sort of fractal matching is interesting, but there's a lot of trivial ways in which that could be true. There's a lot of ways to sort of simulate how people could just be in some sort of Gaussian ballpark. They could be, I mean, so if you flipped the metronome series backwards, you would get actually the same fractal scaling. And so they could be doing it completely wrong. What's more interesting we found was that it's not just sort of them sort of tapping in any order that will satisfy the alpha. what we found more particularly was that there's a particular layout of different fractal scaling exponents within that time series. So you can find a similar sort of multifractal diversity of event sizes with their own temporal structures. And so the range of alpha was actually well mimicked by the human participants. So without any capacity to predict, they are actually engendering the same multifractal structure. And so multifractal structure, I'm abbreviating here and as MFS, and I'm defining that for the time being as the range in alpha. So alpha, you can estimate what's called a multifractal spectrum, and to date, the most prevalently published descriptor of that is the multifractal spectrum width. And so for our purposes, I'm not going to bother you with the other parts of the spectrum. It's the range of alpha that characterizes the width of that spectrum. And so the range of alphas here were better suited in the human participant data showed a range of alpha that matched the original range of alpha in the unpredictable metronome. And it did that better than it matched any sort of linear models of the metronome. So it did not look like this diversity of alphas was reducible to some sort of linear approximation of multifractality in the metronome. Any questions about that? Okay. And so we can sort of like introvert all of that. And so I did some work looking at infant spontaneous kicking. So early in life, a good indicator of sort of a healthy nervous system is some degree of spontaneous kicking, twitching, wriggling. And so I was curious, does spontaneous kicking reveal any sort of exploratory process? There is this story, and has been for about 40 years, that like these early in life kicks and wriggles and stuff is the body learning how to use and explore the gravitational field and the surfaces around it. And so you do get this idea of sort of like a mind fishing for information and casting out and bringing information back in. So I was curious, can you find any sort of relationship in the multifractalities, not between a person and a metronome, but now between different parts of this movement system? And so we had ankle, knee, and hip joint angle information. And so I was curious how those variations would flow upstream. And so in a sense, what I'm trying to do is an elaboration of the metronome study, where instead of a person tapping along with the chaotic metronome, instead of what I have here, or I I was curious if what I had was a series of chaotic metronomes sort of entraining to each other with a bias or an inclination for the flow of multifractality to be upstream. And so what I would see in the data was sort of these joint angles that look like so. And so I wanted to be able to model, to test statistically, like what happens when you increase multifractality at the ankle? Would you get an upstream change in the knee, for instance, and then at the hip? So that upstream flow is what I was most curious about. And so I wanted to sort of figure out how to sort of test that without sort of like squinting at plots and hand waving, basically. And so vector autoregression was a useful tool there, and did in fact confirm that there is this upstream flow from ankle to knee to hip. So what's nice about this vector autoregression approach is that it treats all variables as predictors and outcomes alike. So it allows all these variables to be endogenous. You can fit exogenous variables as well, but I've never been as interested in those, so I haven't really sort of tinkered as much with that. These are linear systems, so the vector autoregression is a linear framework, so it doesn't sort of acknowledge like the potential for something to become exogenous, and so I sort of haven't tinkered a whole lot with that. I've been most interested in sort of like letting all these variables be endogenous and seeing how things interact. And so what this approach does allow is sort of, it allows all, I mean, so written into the assumptions, it allows all items capable, a capability for sending and absorbing multifractal structure, as you might hope if the swarm was sort of fully open and decentralized. Any questions about any of this? Okay, so I'm trying. So basically, what's going on here is I'm sort of trying to broaden the formal foothold for being able to say yes, that there are turbulent flows here where the multifractal structure is propagating through a body. And so as it turns out, anytime we're focusing on any single task, like you can take the motion capture of the point of the body that's doing the task, that's sort of pressed up against the task constraints. So you could be like focusing attention, and that could be your entire job. And you can look at eye movements or hand wheeling of a block. And so, but then anytime you're sort of bringing fractal structure to that direct contact, that sort of hand on the handle of the object that you're holding, or the eye movements against the screen that you're looking at, the multifractal structure implicit in that exploration is riding on this whole body full of multifractal structures. So like it's not that one multifractal spectra actually speaks for the whole body, as we saw with the infant case, and it's possible to extrapolate with the adult bodies, which are more cooperative. We can start sort of estimating how multifractality changes for different parts of these multiple degrees of freedom that had been so challenging to sort of excuse me, shepherd along. And so anytime we're focusing our attention on a task, we are doing so on the background of this big distributed body that's humming with all this multifractal activity. And so these multifractal movements coordinate information across the body. So whatever we're doing in that task and at the part that we're paying attention to, that's sort of in the background of this whole task context and the context of how we're situated and poised in that circumstance. And so we can also look at, and now here's a second organism. We can look at speech and the use of language, and we can show that our perception of language it depends on a meeting of fractal structures. And so in fact, you can look at speech data, so amplitude waveforms of spoken language. And so you can find that, so the air that we expel from our mouth is wonderfully turbulent. It has all this intricate structure. And you can unpack that linguistically in terms of formants and phonemes, and that's really cool. But I'm also interested in how you can look at it in terms of multifractal structure, and it has all this interesting multifractal structure that can then help the listener understand, and then also sort of can engage them as movements, as movement systems participating in the same conversation, as it were. And so it's not just sort of a signal from a source to a receiver. There's a whole full-bodied sort of backdrop for all this stuff, and that's actually not less backdrop and more participative. And so the multifractal movement is actually supporting information transfer in sort of bare bones perceptual terms, but also the abstract linguistic case. Now, so it feels weird, I'll acknowledge, to think about listening as having any sort of movement, because that seems completely passive. But so what you can have is you can sort of give people a listening task where they have to press a button to get the next word that they get to hear, and so they can sort of tap to ask for the next word. And so you can look at that movement time series of button taps to read sort of how engaged the listener is. And that has its own multifractal structure that predicts how well they remember what they heard. So there's actually, so we can quickly sort of generalize from one point of the body to the whole body and then to small groups. And it seems to perpetually, it still seems to be that multifractal structure can sort of predict and support the engagement of multiple different agents working together. Any questions about this? Okay. All right, so here is an actual swarm. So I sort of got tired of sort of waving my hands and saying, yeah, an organism has multiple degrees of freedom and is kind of swarmy. So I started taking organisms that actually were swarms that everyone could agree were swarms. And so we have bee colonies, which are actually the biologists. So I didn't need any psychologists to do this. They called these bee colonies superorganisms. And they've got a lot of integrity to them as a colony without any disrespect to the individual sort of integrity of the individual bees. So there's a hierarchy, but it's suffused with interdependence. So there's role switching without what we can see as direct communication. Individual bees also show quite a bit of independence. They scout for new places, and then they'll campaign to the group for sort of choosing their new places for the next outpost of the colony. So there's group decision making that looks for quorum rather than consensus. So there's actually a lot of nuance. And I'm speaking to a group that is specializing in swarm intelligence. So I don't mean to preach to the choir here. I'm sure everyone here may know a lot more than I do about this. But what I found was a data set where they had individual bee movements measured in 5 different colonies. 2 were enclosed and 3 were open. And so they had little RFID tags. And then they also had basically a card swipe for a glorified card swipe for the bees, where you had an RFID reader appended to the hive. And so every time the bee went in and out, the RFID tag would get swiped. And so there's a timestamp and an identifying marker on each of the bees. So what I found is that sort of multifractal structure of the individual movement is one of the best predictors of colony membership. And so what I tested this in was a neural network multinomial regression that basically took descriptive statistics as well as multifractal spectrum width, and also a t-test comparing that spectrum width to the spectrums for linear surrogates to sort of test how important the nonlinearity of this aspect was. And so it sort of gave me a reassuring existence proof that yes, in fact, it did at least predict. I don't know about cause because I didn't control it, but it's a good predictor of so it's almost as if the swarm might put an imprint on an individual bee's activity. And so it's participating in a group gives it a slightly different multifractal structure than bees in the other colonies. That was sort of the idea here. So this was sort of another step towards sort of finding multifractal structure in swarms and then seeing some sort of indication that multifractal structure did actually co-occur with, if not support, the group coordination. Any questions about that?
Yeah, I have a question.
Sure.
Hi, I'm Franz. So for humans, as you mentioned, like speech patterns, and now you're talking about the bees and their fractal identity tag almost. So the question comes to mind to me is, does the fractal patterns you see in human speech, are they entirely out of our conscious control, or can they?
Oh yeah, I love this question, and I don't if everything else that felt autonomic, like heart rate and all this, if they always seem sort of beyond our control. I have not done the experiments trying to instruct people on sort of training their alphas, but I would love to. And I think that's sort of like a wonderful entailment of these ideas if they hold sufficient experimental water and they are actually sort of doing that job. But yeah, so I don't have any idea as to whether it's explicit. It seems to be moored, as far as we know, into sort of the what could be nicer is like a simpler task context. It's like, do this job, and you don't understand like all the stuff that goes on under the hood. But it's sort of like if you start thinking about like tying your shoes, you can actually get confused because it's become so background. So like but I would love to sort of look at how explicitly we can get people to guide that. So I think that's a great question. Thank you.
Maybe one short follow-up is that aside, the conscious aspect aside, is it more about humans, about kind of like your trained speech patterns, or is it influenced heavily by your current emotional state?
I'm sure emotions have something to do with it. I don't have that information,
Okay.
but I did. But in that study, one thing I didn't tell you about is that I had one condition receive text-to-speech synthesized speech, and so it wasn't a human speaker. And so there was a different use of the multifractal structure. Off the top of my head, I can't remember what exactly that was, but there is that difference was found in so the difference in speaker, human or machine, did sort of pan out to a difference in how people use the multifractality structure in the speech. So yeah, so I think there's a lot of further research to do there.
Well, thanks.
Thank you. So I wanted to get onto slime mold, which so this is an example of another swarm that we wanted to tackle. And so this is a video of a lot of discernible amoeba racing around to aggregation or non-aggregation sites. And so so this is 540 slides of an image taken such that each pixel is between 1 and 3 micrometers. And so you can see that as you already know from your lab's research, these are wonderful organisms that can glom together and then sort of plant seeds for the next generation. And they're about to get together here into their larger structures. I don't think we get to see the full slug here, but we're seeing like a detail of them race into that. So my colleague and I we're interested in sort of how these structures might or might not reflect cascade dynamics. And so we saw a potential to use the spatial series here of the images and do multifractal analysis on that. So you can basically, so it's applying the same principles to time to space now. And so you're looking at pixel intensity in the grayscale. So so does anyone need any primer on Dicty, or can I assume that we all know about Dicty here? Okay. All right. So I'm going to race ahead then. So we have the precision like so. We selected 3 aggregation and 3 non-aggregation sites for each of these frames, and the time in between was about 0.05. Or the sampling rate was 0.05 hertz. So the selections that we picked were squares of 330 micrometers squared. Any questions about those choices? I'm going to come back to this slide, but I want to show you what those slices look like. Okay, so here we had the non-aggregation sites, 1, 2, 3, and the aggregation sites, 4, 5, 6. And so you can see towards the right, as we get through the frames, the bottom 3 have the aggregations happening, and the top 3, there's still amoeba in there, but they aren't coalescing there, and those weren't the hot spots. Any questions about those choices?
So you chose these after the experiment was over? So you let them grow, and then you were like, okay,
yes,
these are the spots where they aggregated, they didn't.
Yes. We did, yeah. So we didn't do this blind. We actually wanted to test post hoc whether or not the cascade dynamic stuff that we've been talking about would find any difference in the relationship across different event sizes. So yeah, so this was pre-cooked, cool. And we got this from the generosity of David Knecht at the molecular bio department at UConn. So yeah. Okay, so we did vector regression of these multifractal dimensions across each of those frames. And so just to jump to the punchline, the fractality of larger events promoted the fractality of smaller events at the aggregation sites, and we got the reverse. So this is sort of analogous to the executive function task with the card sorting, where we got the ultimate sort of outcome, or the task setting there at the outset was showing us different sort of relationships between large events and small events. And so what we're finding here is that the aggregation sites that we picked were characterized across those 540 frames by an exchange whereby the fractal dimensions at higher Q were feeding down on the fractal dimensions at smaller Q, and the non-aggregation sites had basically the small events feeding on the smaller events. And so this was just sort of a nice existence proof again that we just sort of that this seems to be tapping into anything about what's going on. So we don't know for sure, like we haven't manipulated this, so we don't know that it's causal, but it's at least sort of a window to sort of start seeing what sort of swarm-like relationships might be unfolding in this big distributed fluid system. Any questions about that? Okay. So the idea here is that I'm a psychologist struggling with being also, interested in swarms and seeing swarms where other people see single organisms. And so whether or not this is one organism or multiple organisms on the right here, it doesn't necessarily change how we model the emergence of adaptive response to context. So all of these relationships that we'd seen before complicate simpler models, I would say, but then multifractal structure is a nice way to start unpacking what exactly is going on, like how embodiment and how interaction might unfold and how we can start to see it happen. And so cascades are a sort of a geometrical framework to start looking into sort of what swarms we might individually be as humans, and then we can propagate that back to the organisms that we definitely think are all swarms. So multifractality is just sort of an interesting way to operationalize that idea. So that's all I've got. Thank you, and I'm sorry